Well, good afternoon, everyone. Thank you so much for attending this conference that we put together. I'm really excited about being able to share with you some of the work we've been doing on the Cape, as Kevin had just made reference to. So I'm going to be sharing with you today the results of our monitoring trophic interactions in aquatic systems and how this presents a new challenge for citizen scientists. And I will give you some background on that. Before I get started with that, I did definitely wanted to reach out and thank Brewster Ponds Coalition for their support for this project, and also APCC gave us a lot of support in the field and lab support as well, and help with collecting samples. So just a little bit of background on this particular project. This actually started in 2019, and it was done as a collaborative project between UNH and the Brewster Ponds Coalition under the guise of doing research on cyanobacteria. And we were working on Lower Mill Pond, which is in Brewster, Mass. And Lower Mill Pond has a history of problems with cyanobacteria. Sometimes we see the surface accumulations or scums in Lower Mill Pond, and it also has a problem with transparency throughout the summer season. So the project that was started was actually looking at the aerosolization of cyanotoxins in Lower Mill Pond, and you will be hearing more about this tomorrow. We were looking at what was happening with the abiotic variables and biotic variables that could potentially influence the whole process of aerosolization. So the work I was doing was on the biotic variables, and we were looking at the impact of the juvenile alewife population on the zooplankton and cyanobacterial populations in the pond. And the findings from this particular study that the fish populations can affect the plankton and perhaps the production of aerosols as well. So in 2020, we expanded and continued this project that started in 2019. And the objective of the project was whether we could engage our citizen scientists in this process of evaluation to be able to collect and then also analyze the samples that they had collected. And the big question was, what are the most important samples that we need to collect and metrics that we can use to be able to describe the trophic interactions in an aquatic system? So one of the samples that we knew that we needed to use was the edible cyanobacteria, which you will find in the less than 50 micron fraction of the whole lake water sample. So that was our primary sample for characterizing the cyanobacteria. And one of the things that we have to remember about the edible cyanobacteria is that these zooplankton are size-selective grazers. Most notably, Daphnia will be ingesting particles and cyanobacteria that are less than 2 microns in size. Copepods typically are targeting larger forms up to 30 microns in size. The Bosmina will be ingesting particles and cyanobacteria in 20 micron size, and the rotifers are specific for the 2 micron size fraction. So there had been some work previously done in the Haney lab on aerosolization, looking at the different particle sizes and also some of the early work on interactions with zooplankton. So Amanda McQuaid, who was a recent PhD graduate back in 2019, did this really great laboratory based experiment where they were able to document that the presence of Daphnia increased the concentration of particulate aerosols. And Kate Langley, who also graduated 2019, through her field work, was able to characterize what was happening with particulate aerosols and finding that they were mostly in the 2 to 10 micron size. 99% of them are less than 10 microns and 57% are less than 2 microns. So that gives us an idea of what are the size fractions and particle sizes of cyanobacteria and individual cells that we might be concerned with in terms of the process of aerosolization. We also knew that we needed to evaluate the zooplankton communities within Lower Mill Pond. And we had to do that within the context of trying to see what was happening with a classic food web model, which is based on nutrients and energy transfer, and typically represents the bottom-up forces within an aquatic system where we have a positive interaction between the different levels. So starting in early May, the adult alewife in migration begins. And that's when all the things start happening in Lower Mill Pond. And they are starting their out migration in July, early July. So there's a lot going on ecologically in Lower Mill Pond once the spawning happens and the juvenile alewife are maturing. So let's take a look at what happens to the zooplankton in the early parts of the summer in Lower Mill Pond. So we have our size-selective zooplankton. We also have our juvenile alewife, which are planktivorous and typically feeding in the pelagic zone. What we were observing for the past 2 years is by the beginning of June, we see some selection of the Daphnia. So those now have disappeared from the zooplankton community. And by the end of June, typically we are left with only rotifers, which are going to be selectively feeding on our less than 2 micron size fraction. The other thing that we observed in Lower Mill Pond was that the alewife changed their feeding strategy from planktivory to feeding in the benthos. So they are interacting and feeding on the detritus in the benthos. So what does that mean for the rest of the summer when all we have for grazers within our Lower Mill Pond that are the rotifers that are feeding on 2 micron size fraction? Well, if we had our Daphnia present, they have a much higher feeding efficiency than the rotifers do. So the cyanobacterial populations in the less than 2 micron size fraction would be processed much more efficiently. But when they're absent, that efficiency factor goes down, it decreases. And what that results in is basically an excess of small cyanobacteria within the water column. And we observed this by a change in the Secchi disc depth that went from 2 meters down to approximately 0.5 meter. We also were able to see that we changed planktivory from the alewife being in charge of what was going on with the trophic cascade to the chaobarus that are in the system. And it also, the energy that was happening and the feeding on the smaller cyanobacteria is much more important to the microbial loop and what's happening with our flagellates. So what we hypothesize is what's happening in Lower Mill Pond in part is that we're getting an excess of nutrients through internal nutrient recycling, which is now available to the larger cyanobacterial forms. And then we are seeing the surface blooms later on in the season. So what does that mean for the juvenile alewife in terms of them being part of this ecosystem? Well, we know that we have an increase in the amount of small cyanobacterial biomass. And whether that is contributing to the aerosols, we're not really sure at this point how much that contributes to the process of aerosolization, but we are hypothesizing that it does affect it. We also wanted to know what was happening with these alewife in terms of change in their feeding strategy and that they were feeding off of the benthos. So we did some collections, and we took our length and weight measurements of our specimens of our juvenile alewife. And we also looked at their muscle tissue to see if they were bioaccumulating any of the cyanotoxins. So we did observe that the alewife muscle tissue had concentrations of microcystins, BMAA, and anatoxin. And the interesting thing about the bioaccumulation data that we did get was that these concentrations are similar to those that are already published in literature for adult fish. So it's really quite an interesting dynamic that these juvenile alewife, they're just little tiny guys, less than 40 millimeters typically, are having the same concentration and body burden that we're seeing in the adult fish taken from other locations. So the microcystins, we are suggesting that the process of biodilution occurs with that particular cyanotoxin. BMAA, we feel that we observe biomagnification of that. And the anatoxin, we're not really certain at this point whether we are observing biomagnification or whether we're observing biodilution. We also wanted to see if the occurrence of these microcystins in their fish tissue had anything to do with impacting the fitness of these juvenile alewife. And so we used what's called Fulton's condition index, where a value of 3 is considered to be a good value and a healthy population of fish. So our calculated values for that, you can see 2019 was 3.1 and 2020 was 3.18. So what are we doing for this year coming up, and what's happening with citizen science monitoring and trophic cascades? As you can see on the right-hand side, there's already been an article in the local newspaper regarding this issue because of the decrease in the number of herring counts at the herring runs that they have there. So we've decided that we are going to continue our monitoring, and our sampling will include phycocyanin, which is what we typically use for our monitoring program, specifically the less than 50 micron size fraction. And we want to look at not just the concentration of the phycocyanin, but also the contribution of the less than 50 to the whole lake water fraction, because that may provide us more information and relate it to the process of aerosolization. We also want to use some of our zooplankton data this coming year, and we're going to be calculating what's called the predator to panfish ratio using mean crustacean length. And then we also want to use these metrics that calculate a trophic status index based upon the zooplankton populations. And there's many different ones that we can use. We're still in the process of evaluating which ones we want to use, but we are pretty confident that we'll be able to get our citizen scientists involved in at least counting the zooplankton, if not measuring them. So for this summer, what we'll be doing is doing a much larger project on juvenile herring and cyanobacteria interactions on the Cape, and then also out at Martha's Vineyard. We will be doing, with the help of APCC and BPC and the Park Service, and I'm sure plenty of other people, looking at just the cyanobacterial populations on 27 herring runs, where we will be using the less than 50 PC and then the percentage of 50 to whole lake water. We will be doing trophic cascade studies at 3 herring runs. We will be working on Walker's and Lower Mill Pond, which is part of a complex of lakes. We will also be working on Santuit Pond and Herring Pond in Wellfleet. And to this, we'll be doing our phycocyanin data, and then also the predator to panfish ratio and our zooplankton Trophic Status Index. We'll also be doing this out at Martha's Vineyard on Squibnocket Pond. So I'd like to thank the Brewster Ponds Coalition, APCC, Mass Department of Fisheries for their permits, and all of the other folks that have helped out along the way doing this project, and be more than happy to take any of the questions in the chat room.